Hello friends, in this module we are going to talk about the microorganisms which are called archaea. What exactly are archaea? They are neither prokaryotes nor eukaryotes. They show the characteristics of both the bacteria, that is prokaryotes and eukaryotes. They possess genes with recognizable counterparts in the bacteria, showing that the two groups have functional similarities. But then, archaea also possess genes otherwise found only in eukarya. Till recently, the five kingdom classification of Whittaker dominated worldwide, where a very fundamental level distinction was made between the prokaryotic bacteria and eukaryotic plants, animals, fungi, and protists. Hence, it was a rude shock to the scientific community when, in 1970, Dr. Carl Woese found a new group of microorganisms which was different from other bacteria. How were they different? They did not have peptidoglycan in their cell walls. Their membrane lipids had ether-linked side chains in place of the usual ester linkage. The initial amino acid in the polypeptide was methionine instead of formylmethionine. But the most compelling data came from the whole genome sequencing studies, which revealed extensive gene and protein sequence conservation between these newly found members, but not with bacteria. This made Woese to propose that the life be divided into three major domains: Eukarya, Eubacteria, and Archaebacteria, as shown in this figure. He found that Archaebacteria was a misnomer and shortened it to Archaea. Let us now discuss the morphology of archaea. Archaea are tiny, usually less than 1 micrometer long. They range in diameter from 0.1 micrometer to 15 micrometers. Even under a high power light microscope, the largest archaeans look like tiny dots. Fortunately, the electron microscope can magnify even these tiny microbes enough to distinguish their physical features. Some are filaments up to 200 micrometer long. Even with such a small size, archaea show a variety of shapes like spherical, rod-shaped, spiral, lobed, or rectangular. Archaea may be single-celled or may form aggregates. They may be either gram-positive or gram-negative. Archaea may have one or more flagella attached to them or may lack the flagella altogether. The flagella are hair-like appendages used for moving around and are attached directly into the outer membrane of the cell. As far as the physiology of archaea is concerned, they may be aerobic, facultatively anaerobic, or strictly anaerobic. Multiplication can be by binary fission, budding, or fragmentation. They may be mesophiles, thermophiles, or psychrophiles. Archaea may range from chemolithotrophs to organotrophs as far as their nutrition is concerned. They are normally found in extreme environments and hence are called extremophiles. Few are also found as symbionts in the animal digestive systems. Let us move on to talk about the archaeal cell envelopes. As with other living things, archaeal cells have an outer cell membrane that serves as a barrier between the cell and its environment. The membrane surrounds the cytoplasm where the living functions of the archaean take place and where the DNA is located. Around the outside of nearly all archaeal cells is a cell wall, a semi-rigid layer that helps the cell maintain its shape and chemical equilibrium. All three of these regions may be distinguished in the cell of bacteria and most other living things, but when you take a closer look at each region, you find that the similarities are merely structural and not chemical. Archaea build the same structures as other organisms, but they build them from slightly different chemical components. For instance, the cell walls of all bacteria contain the chemical peptidoglycan. Archaeal cell walls do not contain peptidoglycan but have a structure similar to it called pseudomurine. It differs from peptidoglycan in having L-amino acids instead of D-amino acids in its cross-linkages, N-acetyl-tallose-aminuronic acid instead of N-acetylmuramic acid, and beta-1,3 glycosidic linkage instead of beta-1,4 glycosidic linkage. The pseudomurine is hence resistant to lysozyme and penicillin. Gram-negative archaea lack the outer membrane and the complex network of lipids. Instead, they have a paracrystalline S layer of protein and glycoprotein instead of lipopolysaccharides and lipoproteins. This layer may be as thick as 20 to 40 nanometers. Other archaea have additional layers of material outside the S layer. Methanospirillum has a protein sheath external to the S layer. Methanosarcina has a layer of chondroitin-like material. This material is called methanochondroitin. The most striking chemical differences between archaea and other living things lie in their cell membrane. There are four fundamental differences between the archaeal membrane and those of all other cells: chirality of glycerol, ether linkage, isoprenoid chains, and branching of side chains. These may sound like little complex differences, but a slight explanation will make the differences understandable to you. Chirality of glycerol. Let us first revise the structure of the bacterial cell membrane. The basic unit from which the cell membranes are built is the phospholipid. This is a molecule of glycerol which has a phosphate added to one end and two side chains attached to the other end. When the cell membrane is put together, the glycerol and phosphate end of the molecules forming the hydrophilic polar head hang out at the surface of the membrane with the long hydrophobic side chains sandwiched in the middle as shown in the figure. This layering creates an effective chemical barrier around the cell and helps maintain chemical equilibrium. The glycerol used to make archaeal phospholipids is a stereoisomer of the glycerol used to build bacterial and eukaryotic membranes. Two molecules that are stereoisomers are mirror images of each other. While bacteria and eukaryotes have D-glycerol in their cell membranes, archaeans have L-glycerol in their membrane phospholipids. Chemical components of the cell have to be built by enzymes, and the chirality of the molecule is determined by the shape of those enzymes. A cell that builds one form will not be able to build the other form. When the side chains are added to the glycerol, most prokaryotic and eukaryotic organisms bind them together using an ester linkage. The side chain that is added has two oxygen atoms added to one end. One of these oxygen atoms is used to form the link with the glycerol, and the other protrudes to the side when the bonding is done. By contrast, archaeal side chains are bound using an ether linkage which lacks that additional protruding oxygen atom as can be seen in the figure. This gives the resulting phospholipid different chemical properties from the membrane lipids of other organisms. The side chains in the phospholipids of bacteria and eukaryotes are fatty acids chains of usually 16 to 18 carbon atoms. Archaea do not use fatty acids to build their membrane phospholipids. Instead, they have side chains of 20 carbon atoms built from isoprene. Isoprene is a 5-carbon compound and is the simplest member of the class of chemicals called terpenes. Sometimes two glycerol groups are linked to form extremely long diethers and tetraethers of 40 carbons as can be seen in the figure. Not only are the side chains of archaeal membranes built from different components, but the side chains themselves have a different physical structure. The side chains, which are made of isoprene units, are branched off the main chain. See the methyl groups branching out from the main chain. The fatty acids of bacteria and eukaryotes do not have these side chains, and this creates some interesting properties in the archaeal membranes. For example, the isoprene side chains can be joined together. This can mean that the two side chains of a single phospholipid can join together, or they can be joined to side chains of another phospholipid on the other side of the membrane, giving rise to a monolayer. Bacteria and eukaryotes can form only bilayers and not such transmembrane phospholipids, giving rise to lipid monolayers. Another interesting property of the side branches is their ability to form pentacyclic rings as can be seen in this figure. This happens when one of the side chains curls around and bonds with another atom down the chain to make a ring of 5-carbon compounds as can be seen. Such rings are thought to provide structural stability to the membrane since they seem to be more common among species that live at higher temperatures. They may work in the same way that cholesterol does in eukaryotic cells to stabilize membranes. It is interesting to note that cholesterol is another terpene just like that of isoprene. Let us now discuss something about the molecular biology of archaea. Archaea have been found to use eukaryotic-like genes rather than bacteria-like genes for synthesis and repair of DNA, RNA, and proteins. This gene conservation supports the idea of a common evolutionary origin for archaea and eukarya. Let us therefore first try to understand these molecular similarities between Archaea and Eukarya and differences between Archaea and bacteria. The archaeal proteins taking part in the initiation of DNA replication are similar to the eukaryotic counterparts. These include origin recognition complex protein 1, single-stranded binding proteins, primase, mini chromosome maintenance proteins, clamp loader, ATP-dependent ligase, and primer removal protein. On the other hand, archaeal DNA replication is similar to prokaryotic DNA replication in initiating at one origin and proceeding bidirectionally. As far as DNA packaging is concerned, in some members, the DNA is packaged into nucleosomes resembling eukaryotic DNA packaging, but in others, the DNA packing is done similar to that of bacteria. Double-strand break repair in bacteria employs REC-BCD pathway. In contrast, in Archaea, this repair pathway employs proteins homologous to eukaryotic proteins like MRE11, RAD50 and SPO11. In homologous recombination, in bacteria, DNA strand exchange is brought about by REC-A. But in the case of Archaea, this very important step is mediated by an eukaryotic homologue of RAD51 and RAD-A. Translation initiation in bacteria is due to recognition between Shine-Dalgarno sequence at the 5' end of mRNA and the complementary sequence at the 3' end of 16S rRNA. But in Archaea, the recognition is similar to eukaryotic scanning type mechanism. The transcription machinery of Archaea bears a strong resemblance to that of eukaryotic machinery. The similarity includes homologous promoter structure, homologues of data binding protein, TF2B, TF2S, and the RNA polymerase itself. Regarding mRNA, bacterial mRNAs have a short half-life and are thus degraded fast. Archaeal mRNA are more stable, having longer half-lives and thus similar to eukaryote. But on the other hand, like the bacterial mRNA, the archaeal mRNA are neither capped nor do they have a poly-A tail. Before discussing the classification and habitats, let us quickly revise the differences between Archaea and bacteria and Archaea and eukaryotes and also the similarities between them. Both do not have a well-defined nucleus but have a nucleoid in the cytoplasm. Both lack the cell organelles. Both have gas vesicles in the cytoplasm. Both form polycistronic mRNA. Both have 70S ribosomes. In both, mRNA is neither capped nor has a poly-A tail. As far as the differences between Archaea and bacteria are concerned, Archaea do not have peptidoglycan like that of bacteria, but some have pseudomurine. The membrane lipids are not ester-linked like that of bacteria, but are ether-linked. The first amino acid in the polypeptide is not formyl methionine like that of bacteria, but methionine. The tRNAs are without thymidine in the T-cyc arm. Coming to the similarities between Archaea and eukaryotes, both lack peptidoglycan. Both have methionine as the first amino acid in the polypeptide chain. DNA replication and repair proteins are similar. Transcription machinery is similar. Translation initiation is similar. Coming to the differences between archaea and eukaryotes, archaea lack a well-defined nucleus, whereas eukaryotes have a well-defined nucleus. Archaea lack cell organelles, whereas eukaryotes have them. Archaea have ether-linked membrane lipids, whereas eukaryotes have ester-linked membrane lipids. Archaea have 70S ribosomes, whereas eukaryotes have 80S ribosomes. Archaea lack thymine in the T-cyc arm, whereas eukaryotes have it. Archaea have polycistronic mRNAs, whereas eukaryotes have monocistronic mRNAs.